everyone, and welcome to this video introducing RDDC's Mutation Direct tool. In recent years, with the rapid development of gene sequencing technology, an increasing number of pathogenic mutation sites have been discovered. However, there is still a lack of comprehensive and accurate mutation data resources. This efficient and convenient mutation retrieval tool allows for quick searching of reported mutation site information. Furthermore, it can identify unreported mutations and utilize AI tools for splicing and pathogenicity prediction. On the tool's homepage, you can perform a search by entering a gene name, mutation site, or disease name into the search box. We will now walk you through the specific operation process using a case study. For our example, we will use the TP53 gene. The mutation site is C.743G2A, which indicates a change from guanine G to adenine A at cDNA position 743. First, enter the gene name TP53 in the search box. As you type, the system will dynamically display relevant options in a drop-down menu. Select the target gene, and it will appear as a tag in the search box. If you need to filter further, you can return to the search box and repeat the above operation to generate multiple tags for a combined search. For example, continue to enter the mutation site information C.743G2A. After confirming the search tags, click the search button. The search results page will then display all entries related to the target mutation. This can occur because the same gene may have multiple transcripts, meaning the same mutation site can correspond to several results. Click the first nucleotide standard mutation name to access the mutation details page. On this page, the target mutation will be highlighted in purple. By hovering over the information box, you can view the position information of this site and the number of reported mutations. Click the Recorded Mutations and AI-aided prediction button to further view the detailed information of this site, including amino acid changes and pathogenicity reports. In addition, you can use the RNA splicing model to predict splicing abnormalities for the target mutation site. If the target mutation site is not included in the database, you can also click the Patho Predictor button and manually enter post-mutation information to perform pathogenicity analysis. Besides the target mutation, you can also quickly locate and view the mutation status of other sites on this page. If you want to view the amino acid sequence, simply click the Protein Mutations button in the top left corner to switch the view. If you are interested in the transcript sequence, you can click the top right button to switch to the sequence page. This concludes our demonstration of the RDDC's Mutation Direct tool. We encourage you to explore its features and welcome any feedback you may have. Thank you for watching.